Hi folks, this is the second nervous system lecture. This one's basically going to just focus on nervous tissue physiology. So we're going to go through what is an action potential and how do neurons send signals. This picture is just a basic structure of a neuron. A couple things to notice. We have dendrites leading into the cell body. You can see here that the red arrows are showing the direction of a signal. Signals will be coming in through the dendrite, through the cell body, and then we've also got this long projection of the membrane here called the axon. The axon is going to be carrying the signal away from the cell body. In this case, the picture shows a myelinated axon. It's coated with this sort of bluish white covering, and we'll talk more about what that is. And last but not least, the axon has these little terminal buttons at the end, these little rounded projections where it branches off, and we'll talk more about those as well. A nerve is a bundle of neurons. This picture should look familiar to you. You've studied the muscle system, and this is kind of a similar layout. So we have single cells here, one single axon of a neuron, and each cell is myelinated. Those myelinated axons are bundled together and wrapped up with connective tissue, and that connective tissue wrapping all of it together makes up the fascicle. And then many fascicles can be bundled together along with some blood vessels and again wrapped up in connective tissue to create a nerve. Here's a few more vocab terms to add on to this diagram. I've got 6 blanks here and 6 vocabulary terms. So if you remember back to the muscular system, remember that mycium was the sort of end of each of these words. We had epimycium, perimycium. We're replacing that now with neurium because we're talking about neurons. And you should be able to figure out just from the prefix where each of these goes. Remember that the epi, peri, and endo refer to the connective tissue layers. So just take a minute. You can pause your video right now if you want to and try to answer these questions, and then we'll go on. All right, so here are the answers. I've just put the corresponding number next to each word. I actually didn't mix them up at all, it turns out. So they are all in the correct order already. Okay, so if we want to talk about how neurons work, the essential job of the neuron is to send signals. We can see here that this is the end of one neuron. These are sort of the axon terminals of one neuron. Here is the next neuron in the chain. The signal is going to start at the dendrites of this neuron, travel down the axon, reach the axon terminals, and then be passed on to the next neuron. So these are things happening sequentially in a row. We've got one neuron sending a signal. That signal gets passed on to the next neuron. In between each of these neurons where they meet, we have a little space called a synapse. This picture doesn't show the synapse very clearly, but it is actually a space. The two membranes are not touching one another. There's a little gap between the presynaptic and postsynaptic neurons. In this picture, we can see that the electrical signal is traveling away from the cell body down the axon, and we also have a little caption here. This is a myelinated axon. And the idea with myelination is that it helps to speed up signal transmission. So this electrical signal is going to be traveling more quickly down the axon because of the myelin sheath. What is the myelin sheath? It's basically just fatty projections that are coming off of certain helper or glial cells in the nervous system. The central nervous system has cells called oligodendrocytes, and these are shown here where the oligodendrocyte is here, and then we have these branching off, these projections of the cell membrane that grow and wrap around an axon. In the peripheral nervous system, Schwann cells perform a very similar function. We can look at a little bit more of a neuron surrounded by Schwann cells here. And in this case, the membrane of the Schwann cell is all this purple stuff, and it's surrounding the axon. One Schwann cell is basically going this distance, and then we have another Schwann cell here. And in between, there's actually a space. That space is called a node of Ranvier. The little gaps between the Schwann cells are going to be the only places where the axon is exposed to the extracellular fluid, and that's important. We'll talk about why a little bit more, but just keep in mind that we have multiple cells wrapping up the length of the axon with little gaps in between. All right, so how does the neuron actually send a signal? The signal that the neuron sends is going to be called an action potential, and that action potential will start sort of here towards the base of the cell called the axon hillock, and it's going to travel down the length of the axon. It's going to travel from the cell body towards the axon terminals. And what's really happening as the signal is being sent is that the axon is depolarizing. What does that mean? Normally, a neuron at rest has a polarized membrane. And all that means is that the inside and the outside have different charges. The inside, in this case, is more negative than the outside. This just happens because of an imbalance of ions across the membrane. This is the neuron at rest is going to have more sodium outside and more potassium, which are the little green dots inside the cell. The inside also has lots of other negatively charged particles like amino acids, and so it has an overall charge that is more negative than the outside. The action potential is actually going to be the axon depolarizing little by little. So the action potential is going to be where one region of the axon depolarizes, and that's going to trigger the next region to depolarize, and that's going to trigger the next region to depolarize. We'll talk about what that means in just a second. Notice the diagram though here, we have these little red arrows sort of jumping down the axon, and the depolarization is really just happening here in these nodes. The name for that is saltatory conduction, where we have the signal just sort of jumping down the axon, not traveling all the way, not having to depolarize at every single spot along the axon, but just within the nodes. And that's how the signal is spread more quickly. So we've said that a resting cell is polar because there's a charge difference across the membrane, and to depolarize the cell literally means to take away the polarization or to even out the charges so that the inside of the cell becomes more like the outside of the cell. What actually happens is that sodium ion channels open up, and that allows sodium ions to move into the cell. Remember that the inside of the cell was negative, and we can see here sodium ions are positive. So if we're having positive ions flowing into our negative space, it's going to kind of neutralize the inside of the cell. The inside of the cell becomes more positive. Okay, here is saltatory conduction again. This is just, again, the depolarization is happening at the nodes. So you can see here that the nodes are actually where the sodium ions are going to be able to pass into the axon. The little gates that let them in are found only at these nodes. Once they enter the cell, though, they can kind of diffuse along and move towards the next node. The important thing about this action potential and how it actually gets propagated or how it gets continued all along the axon is that depolarization of the axon in one place is the trigger that causes depolarization at the next node. So depolarization here, the rushing in of sodium atoms, is going to result in more sodium gates opening up at the next node, which is going to then result in depolarization at the next node. So we can think of this kind of like a row of dominoes where one triggers the next, until we get all the way down to the end of the axon, to the axon terminal. Just to show this a little bit more clearly and to help you kind of visualize what's happening here, I have a video link for you on YouTube. I'm going to walk you through it. There's no sound on the video, so I'll just sort of talk you through what's going on. So here's our video, and we can see that this is a resting neuron. Up top here is telling us what phase we're in. The cell is at resting potential. We can see a cell membrane with some ion channels. Here's a potassium channel and a sodium channel embedded in the membrane. We can also see over here what's called a sodium-potassium pump. If we were to put signs for charge in this picture, we would have a negative inside the cell. The inside of the cell at rest is negatively charged, and the outside is positive in comparison. So I can hit play here. Oh, actually one last thing. Notice that inside the cell at rest we have lots of these purple potassium ions, and then on the outside we've got a lot of sodium. So at rest we have more sodium outside, more potassium inside, and the inside is negative. So depolarization, the sodium channel opens up and sodium rushes into the cell. Keep in mind that these little sodium guys are positive, so as they're rushing in, they're neutralizing this inner area. That's going to be called depolarization because we are going from a polarized membrane where there's a big difference in charge to a less polarized membrane where the charges are evening out a little bit. The next thing that's going to happen is repolarization. The potassium channels open up and potassium leaves the cell. Potassium is positive too, so when the potassium leaves, the inside of the cell becomes negative again. We've reset up that polar resting potential. We've reset up the charge difference across the membrane. Now the process isn't entirely finished because if you notice where the ions are is not where they started out. So a resting neuron needs to have potassium inside the cell, sodium outside the cell. And that's going to be this last phase here, returning to resting potential, where the sodium-potassium pump is actually going to use ATP to push the ions across the membrane into their appropriate spots. We need to build up a high concentration of sodium outside and a high concentration of potassium inside. And once that happens, we are back to our resting potential. This is where I'm going to stop this video. You're going to see the rest of it on the part 2 of this video lecture. So we'll talk more about this graph. Just click on